Hi, my name is Emily Oldani, and I'm a postdoc researcher over at the University of Denver. I was just funded through the Movement Disorders Foundation Young Investigator Pilot Grant to do a project entitled A High Throughput Cellular Approach to Identify Potent Antagonists of HTT, or Huntington Aggregation, Mediated HD, or Huntington Disease Pathology. So I'm just going to give you guys a little bit of an overview of the research that my lab does, as well as what the aim of this project is. So make the really long title pretty digestible. So my background is as a biochemist, so think more test tubes in a lab space than interfacing with patients. So I approach this problem of neurodegenerative diseases, so it's going to be the aging diseases, from looking at protein or a biomolecule aggregating or clumping up, which causes neurodegenerative and neurologic diseases. So in a healthy brain, you have protein, these biomolecules being constantly produced and existing and performing a role in monomers, which are just going to be one copy of that biomolecule. Those monomers can come together to get oligomers, which are going to be some of those biomolecules, not a lot, so fewer than a lot, more than one copy of them. And that's still often pretty functional for how they're functioning in a cell and able to perform their jobs. But sometimes they can start to go off pathway from what their goal was and what their job in a cell was and in a brain, and they'll start to form these protofibrils that start to clump together to form amyloid fibrils and those plaques that you've often heard of referred to in diseased and damaged brain tissue. So you get these holes punched through tissue, you get these unclearable biomolecule clumps, and our lab and my project aim to stop that. So we want the biomolecules to not clump through the use of our drugs that we design, synthesize, and test all in-house, to maintain and keep those biomolecules from clumping so that we stay in just that healthy tissue side rather than the diseased tissue side. So aging and neurologic diseases are when you go from having a healthy brain to a diseased brain through cellular death, and that cellular death can often be caused by those biomolecules clumping, which can be influenced by genetic factors or environmental factors. Environmental factors are, for the most part, going to be any sort of stress caused onto cells that could be from breathing in toxins, they could be from negative health impacts, they could be from traumatic brain injuries, they could be just from aging itself, and that builds up over time, and cells aren't able to take care of themselves as well. Four of the main ones that our research specifically goes towards are Alzheimer's disease, Parkinson's disease, amyotrophic sclerosis or ALS, and Huntington's disease. Specifically with Parkinson's disease, we have two drugs that we have designed already gotten to the point of being in mouse models. So we've successfully been able to target the biomolecule clumping in that and prevent it from happening in a variety of tests. We have also started designing a way to make the process for this go faster so that as we move towards our Huntington's work, we can start getting hit molecules that are successful in preventing that biomolecule clumping and maintain healthy brain tissue instead of diseased brain tissue to go a little bit quicker. With drug design and any sort of work with that, you're trying to balance two things constantly. You want something to both be small, because if it's small, it can get into cells and it can get to your targets, in this case, those clumping biomolecules, really easily. However, you also want it to be specific. You want to know that your drug is doing exactly what you think it is and only interacting with what you want it to interact with. You don't want it to cause all sorts of weird effects through every cell and every tissue in a person or other organism. So we want to try and balance small and specific. So on one hand, you have small molecules that are too small and not specific at all, but they can really get into cells. On the other hand, we have too large, so antibodies, they're going to be highly specific, but they're too big, and our drugs fit nicely in as being more towards the small scale and just big enough that they're pretty specific to the biomolecule targets that we select and identify, so they're the best of both worlds. There's a general therapeutic pipeline that our lab follows, where we start in HEK cells, which is a kind of human cell that's very easy to culture, and you can quickly move forward through checking if the addition of a molecule is promoting the health of a cell, not making a cell more sick, and that it is actually able to interact with your target, so keeping those biomolecules from clumping. If something is successful in that, we can bring it forward towards primary culture neurons, which just means I'll have neurons that are in a dish that I'm working with, so they're not in a brain, just in a plate. If something's successful there, we can move towards C. elegans, which are a flatworm that's very easy to monitor the lifespan of, because it's going to be on the scale of weeks rather than months, years, decades. And they're very sensitive to aging and neurologic diseases. If something's successful in that, we are able to then move it towards mice. So, and I'm sorry, my head is kind of obscuring where an arrow is right now, but an orange arrow just popped up saying this is where our Parkinson's model currently is. It's at the mouse phase. And our Huntington's model is currently at the phase of being able to test our drugs in cells. So generally, there's a therapeutic pipeline, which is how you go from having an idea to having an actual therapeutic that works. As you can see, moving from left to right, you have a broader spectrum of drugs that you're looking at that starts to narrow and get down to probably just the one, if it even makes it that far. So you start with your target hypothesis, you have a lot of ideas, a lot of things to test, and that phase starts to happen pretty quickly. As you start identifying what ones work, what ones don't, and can further refine those, you get to a hit, a lead, a candidate, and then you start getting towards moving towards in people and getting like FDA approval. So a hit, that's approximately where the cellular model starts for us, and then the preclinical and candidate location in this graph is where you would have the mouse model coming in. Another thing to take note of going from target hypothesis to submission to launch is time is going to increase. So the first steps are going to happen very quickly and the second steps very slowly, which is why we'd like to further develop an assay we've started with that speeds up a little bit of that change from hit to candidate. Currently, for Parkinson's and Lewy body dementia, we have molecules that are at the phase of being in mice, so they're preclinical. And we're further characterizing those as well as using them as a known way to test to make sure that our speeding things up test does work. Initially, we have a library already of drugs to test with the Huntington's project, so it's already starting at the location of identifying a hit and selecting that. So we have drugs that we're going to add to cells that we were just able to buy with this funding from the Movement Disorders Foundation. The overall goal of this project, as well as the lab that I'm a member of, is to prevent this diseased and decaying with plaques and biomolecules clumping up, dying neurons, aging and neurologic disease tissue from happening in brains, through the addition of our drugs to then rescue it towards this healthy brain tissue again. So this can either come from using our drugs to prevent it from ever getting to that disease state, or the addition of our drugs to rescue it back to recovered tissue by allowing the cells to have time to heal and regrow and no longer have those plaques added into them. With that, I'd like to thank the University of Denver, as well as the Noble Institute of Healthy Aging at the University of Denver, the Movement Disorders Foundation, the NIH, Parkinson's Foundation, and then the American Parkinson's Disease Association, as well as the Department of Defense for their generous funding. I would like to thank my PI, primary investigator, or boss, Dr. Sunil Kumar, as well. I'd also like to thank a collaborator that has worked with the Movement Disorders Foundation before, Dr. Daniel Paredes, and all of the current and past graduate and undergraduate students in our lab. Thank you so much for your time. I really appreciate that, and I look forward to giving you guys updates on this project in the future. Thank you so much.